The following are instructions for using the NANSLO lab interface for remote web-based lab activities using the microscope. The equipment control software shown here is written using the LabVIEW software from National Instruments. The user interface is presented as a LabVIEW front panel, which will be referred to as the lab interface for the remainder of this training. This interface, used for NANSLO microscope activities, allows you to focus samples on the stage by moving the stage up and down, moving the stage forward and backward on the y-axis and left and right on the x-axis, adjust the light intensity using luminance, select from several objective lenses with different magnification capabilities, use various color modes to enhance the sample image, view a real-time video feed of the sample on the stage looking through the microscope in a display window, capture a high-resolution image of the sample and save it to your computer, or copy it into a document. Through the picture-in-picture feature, you can view different locations within the lab where the activity is being performed. For example, one camera preset allows you to view the objective lens and the action of focusing. Use 6 different preset camera positions to view these different locations. Pan the picture-in-picture camera left to right and/or tilt it up and down. Zoom the picture-in-picture in and out. When using the arrows on this control panel, click and hold the button until the desired effect is achieved, or click and wait to view the result before clicking again. Quick clicks on the arrows may cause the system to lock up. When multiple students are using the microscope at the same time, right-click anywhere in the gray area of the control panel and choose Request Control of VI from the dialog box that appears. After you request control, you may have to wait a short time before you actually receive control and are able to use the features on the control panel. If you are not certain if you have control, right-click again. If you see Remote Panel Client, then you have control of the microscope. Click anywhere in the gray area to close this menu. To release control of the microscope, right-click anywhere in the green area, select Remote Panel Client, and then select Release Control of VI. The controls for the remote web-based science lab microscope are very similar to controls found on an on-site microscope and behave the same way. We will first discuss the stage controls. These are found in the upper left-hand corner of the lab interface. Samples are placed on the stage for observation. 6 buttons control the direction of movement. Through the lab interface, you can move the specimen in both the X, which is right to left, and Y, which is backward to forward directions, so the entire sample can be examined. These arrows allow you to precisely control the position of the sample on the stage. Use the backward and forward arrows to move the stage to view the top, middle, or bottom of the specimen. Use the right and left arrows to move the stage to view the specimen from left to right. Note that when you click on the forward, backward, left, or right arrows, the sample appears to move in the opposite direction. Since the objective stays fixed, the image moves in the opposite direction of the stage. This is how these controls work on most microscopes, so the feel of the microscope is preserved over the web. the Z direction, up and down arrows, move the stage closer or farther away from the object lens to bring a sample into focus. When selecting the button between the two arrows, you can toggle between coarse and fine. When the button is dark green and coarse fine is displayed, to the right of the button, the microscope is in coarse focus. When the button is clicked and turns light green and fine is displayed, the microscope is in fine focus. Typically, you will start with coarse focus, which moves the stage in large increments. Next, you will change to fine focus. It moves the stage in smaller increments. Note, there is no difference between the coarse and fine focus when using the 60x objective. This microscope contains parfocal lenses, so once an image is brought into focus through one objective lens, it will be in focus when another objective lens is selected with just minimal fine-tuning. After clicking the Automatic Focus button, the microscope will focus automatically. You will see the image of your specimen go out of focus and then come back into focus. At times, you may have to press the button several times to get the best focus. If you wish to focus on different planes within your sample, then it would be better to use the up, down, and coarse and fine stage controls rather than relying on autofocus. The luminance dial determines the brightness of the light illuminating the sample and can be set between 0% and 100% brightness. For this control to work properly, you must have the automatic exposure button turned off. Otherwise, the camera will automatically adjust the exposure and defeat your efforts. By default, auto exposure is on. To brighten the sample, begin with the automatic exposure button turned on and turn down the luminance control until it reads close to 0. You can do this by holding your left mouse button down and moving the arrow towards 0, or you could also type a number in that box. Wait for the camera to automatically adjust the exposure. Then turn off auto exposure and adjust the luminance control upward until you get the illumination you want. To reduce the illumination, begin with auto exposure turned on and adjust the luminance control up. Wait for the camera to automatically adjust the exposure. Then turn off the automatic exposure and adjust the luminance control downward until you get the illumination you want. A microscope mounts an objective lens very close to the object to be viewed. Depending on need, different lenses with different power will be used on the microscope. This microscope features multiple objectives, each with different power, mounted on a rotating turret. The larger the magnification numbers, the greater the magnification. For example, if a specimen is viewed through a 40x objective lens, the magnifier in that lens displays the specimen 40 times larger than an equivalent view as seen by the unaided eye. Remember that the ocular or other lenses also add magnification. This microscope has 5 lenses: 4x, 10x, 20x, 40x, and 60x. Use the arrows below the objective lens box that indicates the magnification of the current objective lens to move to a higher or a lower magnification lens. If you have activated the Picture-in-Picture preset, you will be able to see the objective lens move when you select a new magnification. The camera control unit is equipped with a series of image processing functions that enable you to quickly and easily correct imaging problems that arise from low or high contrast, poor focus, insufficient or uneven illumination, sample shading, or discoloration and noise. The most common reason for uneven illumination is a light source that does not completely fill the field of view on lower magnifications. A choice of color modes can be selected in the microscope image area. The default setting for this feature is normal. In the normal mode, the sample is displayed its true color. In the negative mode, the sample is displayed in a color-inverted form, where red, green, and blue values are converted into their complementary colors. The technique is useful in situations when color inversion can be of benefit in exposing subtle details or in quantitative analysis of samples. In the blue-black mode, the black portion of a grayscale negative sample is displayed in blue. This mode is often used to reveal details in samples having a high degree of contrast. The blue-black filter can aid you in examining a wide spectrum of difficult samples. In the black and white mode, a grayscale image of the sample is displayed. In the sepia mode, a brown scale, black and white image of the sample is displayed. Although typically this filter is of little utility, it can be employed to alter image color characteristics to improve the visualization of sample detail. At times, the sample may have an unacceptable color quality. Use white balance calibration to remove the color cast. This process is often referred to as white balancing. Auto exposure is automatically selected. You do not need to do anything with auto exposure unless you are adjusting the luminance. If you are doing so, you should turn off auto exposure. The camera on this microscope allows you to capture and save the microscope image. When the Capture Image button is pressed, a 5-megapixel image is opened in a new tab in your browser. When the image is being captured, the button will illuminate light green. The capture is complete when the light turns off. Be patient, as this may take several seconds to complete. After the capture image light turns off, select the View Captured Image tab on the bottom of this control panel to view the image. If you want to magnify into a specific area, you notice that you have a magnifying glass. You would just select your left mouse button and it will further magnify in the area of interest. To save this image, first right-click, then notice you have two different options here. I'll step you through both of them. The first time you right-click to save an image, you will be prompted by a Citrix receiver security warning like the one shown here. It is important that you check the appropriate box and correct access. For a Windows operating system, select Yes and check the Do Not Ask Me Again for this site box. For a Mac operating system, check Don't Ask Me Again and then select Read and Write. It is recommended that you rename that picture, otherwise you may overwrite a picture that you have already saved. So I am going to call this image 2. Also remember that when you are saving the image, be aware of where you are saving it on your own computer. In this case, it is being saved in Libraries, Pictures on my computer. Click on the Save button, and the image has been saved. Another option that you have, if, for example, you're doing a lab book and are using Word to create your information, you could also right-click, select Copy, go over to your Word document, and select Paste, and that will paste the image into your document. The Image View window displays the real-time video feed from the digital camera looking through the microscope. All of the controls below the Image View window are applicable to the Picture-in-Picture feature. To activate this feature, first select the Picture-in-Picture button. A second real-time video feed from another digital camera appears in the Image View window. There are 6 camera preset positions. At this time, number 1 is selected. Preset 1 displays the microscope, microscope camera, and a camera control unit projecting the sample on the stage. Preset 6 is the same as Preset 1. When you select Preset 2, you see a close-up of the objective lens. When you select Preset 3, you see a close-up of the camera control unit projecting the sample on the stage. When you select Preset 4, you see the microscope eyepiece and the camera mounted on the microscope. And when you select Preset 5, you see the condenser lens underneath the stage that focuses the light on the sample. The condenser lens controls the width of the beam. In some instances, you will want a tighter beam, while in other cases you will want a broader beam to control the image quality. Currently, this setting has been optimized for you. However, in the future, a control may be available to select the optimal setting for your sample. For each camera preset view, additional camera options are available. Use the up arrow to tilt the camera up. Use the down arrow to tilt the camera down. Use the right arrow to pan to the right. Use the left arrow to pan to the left. The camera also has zoom capabilities. To zoom out, select the Zoom Out button. To zoom in, select the Zoom In button. These instructions were produced by Western Interstate Commission for Higher Education. For more information about NANSLO, please visit www.witchy.edu/NANSLO.